Parkes Disease Test Team. Welcome everyone, and thank you for joining Parkes Disease Task Team webinar, B-Cell Basics, Better Together. My name is Michelle Chrisman, and I'm with the U.S. Fish and Wildlife Service, and I'm the Federal Agencies Coordinator for PARC, Partners in Amphibian Reptile Conservation. And with me is Carrie Wickstead of the Association of Fish and Wildlife Agencies, who's the state coordinator for PARC, and she's going to be behind the scenes managing tech for the webinar today. So we are pleased to be partnering with the North American B-Cell Task Force to bring you the third in PARC's webinar series today with a presentation on the basics of B-Cell and why we should care, as well as how we can work together to proactively prevent, detect, and respond to this disease threat. So our goal with the webinar series overall is to help connect science and management action by providing information to land and natural resource managers. And so we welcome input and feedback after this webinar as well. And then before I introduce our speakers today, a little housekeeping. You should see closed captions enabled and subtitles on your screen. You can turn that on and off by clicking the CC button down at the bottom. And then this webinar is also being recorded for later posting on the Park Disease Task Team webpage and the Park YouTube channel. And all attendees will be muted until the Q&A session at the end. Questions can be asked via the chat during the presentation. And then with us, we have Dee Dee Olson, a research ecologist with the U.S. Forest Service, and Jake Kirby, chair of biology at the University of South Dakota. And they're both former chairs of the B-Cell Task Force and involved with some of the working groups. And they'll be monitoring the chat for questions that they may be able to answer. And then we're also going to be collecting questions for the Q&A session at the end. So Carrie and I would like to thank the Parkes Disease Task Team for supporting the ongoing amphibian and reptile health webinar series, as well as the North American B-Cell Task Force for helping to develop a variety of the products mentioned in this talk. We would like to especially thank our two speakers today, who are the co-chairs of the North American B-Cell Task Force. And so with that, I'm now pleased to introduce our speakers today, Dr. Molly Bletz and Dr. Evan Grant. So Molly is a researcher at UMass Amherst working to understand B-cell disease risk, to develop integrative disease management tools, and to optimize management decisions for B-cell. She is also the 2021-22 chair of the B-Cell Task Force and serves as the director of international disease mitigation for the Amphibian Survival Alliance. And Evan is a research wildlife biologist with the U.S. Geological Survey's Eastern Ecological Research Center and a principal investigator of the Amphibian Research Monitoring Initiative. He conducts research on amphibian populations, their ecology, threats, and management to support federally protected parks and refuges. And he's the chair of the North American B-Cell Task Force for 2022-2023. And so with that, I will pass it over to Molly to start the webinar. Take it away, Molly.
All right, well, thanks, Michelle, for going over those logistics and the introduction, and welcome everyone. We're really happy to have you all here today to provide an overview about this new chytrid fungus, B-Cell, and the North American B-Cell Task Force, and sort of the who's, the what's, the where's, and the how's of this pathogen, in addition to the task force itself. And we're really excited for the opportunity to be able to address any questions that you have throughout this webinar, and also time to discuss how the task force can better serve the needs of managers and the broader community. Hopefully I will be able to advance here. All right. So Batrachochytrium salamandrivorans, or B-Cell, is a chytrid fungus that's pathogenic to amphibians and was first detected or described in 2013. The earliest occurrences are now as far back as 2004 in Europe. It's related to BD, or Batrachochytrium salamandrivorans, which was described in the 1990s. And wreaked havoc on amphibian populations in regions of the world like Central and South America, Australia, in the Western US, and still is a threat in the Western United States to species like the mountain yellow-legged frog. B-Cell, like BD, co-evolved with amphibians in Southeast Asia. excuse me, but was recently introduced to Central Europe and is causing devastation to their natural amphibian communities in places like Germany, Belgium, the Netherlands, and a recent outbreak that's happened in Spain. And so salamanders appear to be the most affected by B-Sal, with European fire salamanders, as you can see here, experiencing the most striking declines, and populations really being driven to complete extirpation or really, really low densities. And so this is particularly concerning for North America, given we have a really high and unique diversity of salamanders. We have over 50% of the world's salamander diversities within the US alone. So there's been a large effort, a lot of which has been led by Dr. Matt Gray's lab at University of Tennessee, to understand susceptibility through lab-based experiments. So far, 55 species have been tested, and 76% of those were able to be infected with B-Sal. And that includes species across the diversity that's present in the US, from newts to mole salamanders to lungless salamanders, and even frogs. And 35% of those tested species developed disease and experienced mortality in the lab. And importantly, both salamanders and anurans and frogs develop disease and experience mortality, which is different from some of the original work that was, which is like an additional piece of knowledge that has been gained as work continues. And the species genera we have listed here, including different types of newts, plethodonid salamanders, and some frogs, including spadefoot toads and Cuban tree frogs, are those that experience mortality in the laboratory trials. And so why the ones we listed here are the ones that seem to be the most susceptible to B-Sal-induced disease. It is important to note that lab experiments don't necessarily 100% equate to what's going to happen in natural systems. And so different disease dynamics might occur in natural systems compared to these trials, with species being more or even less susceptible in natural ecosystems. But the important take home here is that there's multiple and many North American species that are highly susceptible, including frogs and salamanders, that can be potential carriers and negatively affected by this pathogen. So B-Sal does its damage by infecting amphibian skin. It has swimming aquatic zoospores that, once they find an amphibian host, can burrow into the skin. And as that infection builds across the skin, it can result in multiple clinical signs, including things like skin shedding or blistering and ulceration of the skin, emaciation, ataxia, and ultimately can lead to mortality. The severity of these symptoms, of course, and what types of symptoms happen is dependent on a lot of factors, including which species is infected. They don't all present the same disease symptoms. It can vary with life stage or different environmental variables like climate and water quality. And one important piece of these symptoms is that they aren't really unique to B-Sal. A lot of amphibian diseases show very similar symptoms, for like skin shedding, for example. We can point to sort of the current COVID pandemic for some parallels here. And I recently just got over COVID, and my symptoms were very much like a common cold, but we of course don't want to dismiss that before we know what it is. And so likewise with amphibians and B-Sal, we don't want to be dismissing die-offs or sick amphibians that have symptoms. And it's really crucial that when we see something or encounter a die-off or a sick amphibian, that it gets reported and tracked and followed up on to understand what might have caused it. Okay, so B-Sal is a pretty crafty pathogen, and we're still learning lots about how this pathogen moves about. Introduction and transmission between sites can occur in a lot of different ways. We might think first to animals and amphibians moving B-Sal across the landscape, and that can happen both through natural movements related to migration events or dispersal. Birds have been proposed as a potential source of movement because the pathogen pathogen can sort of embed and insist on the keratinized portions of their feet, which sort of gives this avenue for longer distance dispersal. And there's also a human-mediated animal movement that could contribute here, for like accidental pet release, if somebody has a pet frog that escapes or is released to the wild, or bait related release. In certain regions, salamanders are used as bait. And so if at the end of the day you dump your bait somewhere, that could potentially be spreading disease. And this human-mediated movement side has opportunities for intervention through policy or different regulation efforts, which is one step that has occurred in the United States, and there's similar efforts that have happened in Canada related to sort of minimizing the introduction risk for this pathogen by the interim ruling in 2016 that basically used the Lacey Act to list over 200 salamander species as injurious, which prevents these species from being imported into the United States. And this includes basically any species or genera of salamander that is known to carry or be able to be infected by B. sal, includes species from Europe, from Asia, and also from North America. And so breaking these pathways of introduction is really valuable for minimizing risk of B. sal arrival. But these bans, of course, have limitations. As you remember from a minute ago, B. sal is not limited to salamanders, and frogs can be carriers of this pathogen. And so recent research has shown that Cuban tree frogs, which are invasive in the southern United States and also commonly traded as pets, actually get infected and can develop chytridiomycosis from B. sal. There's also work by Anita Tavares and others that have found that highly traded species like this Latoria, which is an Australian white tree frog, or Hymenocyrus, which is the African dwarf clawed frog, also can be infected by B. sal, and these species actually don't show any external symptoms or signs of disease, making them these sort of potential silent carriers of the pathogen in the pet trade. There are efforts underway through partnerships with the Pet Advocacy Network and different pet industry representatives to find ways to minimize B. sal risk while maintaining sort of healthy and clean business operations. So we're talking about animals sort of moving these pathogens around. There's also other ways like fomites and environmental substrates that can contribute to introduction and site-to-site transmission. This could happen through natural flow of water, or again, through human-mediated movement of fomites in soil or leaf litter. Maybe something gets stuck in a boot or on a piece of equipment that's being used in the field, and then tracked between sites. There's recent studies that show that some strains of B. sal can sort of live and sustain themselves on beech leaves, which basically just provides an opportunity for the environmental persistence of B. sal to be longer. And if it's persisting in habitats outside the amphibian, can be a lot easier to move it around. Fire salamanders, so research out of Europe has shown that fire salamanders can be infected with B. sal for up to 3 days from contaminated soil. And so it doesn't have to be direct host-to-host contact for that transmission. So this fomite-related movement highlights the need for biosecurity measures like boot and equipment disinfection that we can all do when we're out in the field. There is a resource that the Northeast Park Group has been working on that brings together disinfection protocols for not only B. sal, but BD and ranavirus, which should be coming out and being published shortly. Okay, so we've been talking about that movement between sites or introduction into a new site, but there's also a lot of patterns and dynamics that are going to happen within a given site with transmission between amphibian hosts, between individuals and different species. And this also depends on many different things. It depends on what species are present there. Do you have a lot of species who are resistant to being infected, that are tolerant, that are highly susceptible to the disease. Host density can play a role in the fact that if you have more density, you're going to have more contacts between individuals. Environmental conditions are really important, like temperature and salinity. B-Sal is a cold-loving fungus. It can tolerate temperatures up to about 25, but then it becomes very, very unhappy. And habitat complexity is another thing that recent research has shown to be an important aspect of that within site transmission dynamics, with the idea being, or the concept being, that they're increasing complexity or woody debris habitats or aquatic vegetation can reduce contacts between individuals and in turn reduce that direct transmission. Building understanding of all these different factors can also shed light on what factors we might be able to manipulate as a management action to allow populations to persist through B-Sal. And so can we increase habitat complexity through adding woody debris or putting some plants in the pond to reduce transmission? Can we increase temperature at certain times of the year to limit pathogen survival? Can we introduce micropredators? So in the image over here, you see this predatory zooplankton. There's little micropredators that swim around in ponds and can eat the fungal zoospores. And so we could increase the abundance of those communities to reduce the infectious B-Sal spores that are swimming around and minimize infection potential for the host. And the answer to many of these things, like in many times in ecology, is it depends. There's, of course, a lot of different things that affect dynamics that have to be considered. And there's, of course, more than just B-Sal and the amphibians that managers are going to be considering as they make management decisions. And so these management actions and what the best action is going to be context dependent. And we'll be coming back to that as we talk about the task force in the second section. And so all of this information that we've learned about B-Sal, we've learned a lot about the biology, we've learned about the ecology, host susceptibility, environmental tolerances, as well as implemented some policy actions to help minimize risk. But these can all inform risk models like the one we're looking at here, which is by Greer and colleagues in 2021. This risk assessment is including information about North American species susceptibility, climate conditions of different regions, and also incorporating those trade dynamics of the volume of imports and now the volume that isn't coming in from salamanders. So incorporation of the fact that that salamander ban with the Lacey Act is eliminating entry of certain species. And while the salamander rule does help reduce risk, we can see that there is still a high risk in a lot of areas across the US, most of which is in the eastern US and the western and the west coast. So we've learned a lot since B-Sal was first described, and preventing entry and completely eliminating risk, introduction risk, is pretty much impossible, which is why there's still a lot of work to be done for this pathogen threat. And it's crucial that we work together and to prepare for and proactively manage for this B-Sal threat. And with that, I think we are now going to break for our first question period. So if you have questions on the sort of specifics of B-Sal or the information we've covered so far, please feel free to, you will have the ability to unmute now, but please raise your hand so we can sort of coordinate discussion, or if you're more comfortable, you can type your questions into the chat as well. The next section will be talking about the North American Task Force and how we can work together so we can save questions related to that until later on.
So Molly, there was one question in the chat that we can open this with. It's from Alex Wolf, and I'll just read what he wrote. How to identify a die-off event versus a few natural deaths? How many dead is a die-off? And how to diagnose a die-off as B-Sal versus other pathogens or other causes like climate weather events, for example, a late freeze in the spring?
Yeah, so you're highlighting a really great point that amphibians can die from a lot of different reasons. And I guess to talk, we do have a rapid response template, which we'll be talking a little bit about in this next section, that does highlight some of the ways to sort of work through those decisions and with information that you do know and information that you don't know. If you have the ability, for example, to collect individuals, those can be sent to a laboratory who can look for evidence of pathogens like B-cell. There's also swabbing that can be used to detect B-cell, but that protocol really outlines sort of the decisions you can make along the way for sort of disentangling what to do if you see dead amphibians. There's also the herp disease alert system, which maybe I will sort of pass to Dee Dee if you want to say a quick note about that.
Yeah, it was a kind of a multi-part question. Let me just, if there's a die-off, and I think it's a good idea to pursue any die-off, so it doesn't matter how many animals that you are finding. It would be good to know what if there is a disease cause of that die-off. And for B-cell, we would require animals to be sampled at 2 independent laboratories to confirm the presence of B-cell. And that in a few minutes, Evan will show the rapid response template, which is a document that gives the outline of how to respond to a B-cell die-off. And that's at the salamanderfungus.org website as well. Yeah, if there's a die-off, if there are dead animals, we'd like to know if it's a disease cause, and then if it's B-cell, that triggers a whole bunch of responses. And Molly, what were you throwing my way? I forgot already.
I was just thinking of the herp disease alert system and
that
connecting. So if you are not somebody who works for the state and you saw a die-off, sort of that avenue as a point of connection was.
Yeah. So the Park disease task team initiated sort of the, Ghostbusters, who are you going to call if you find some dead things out there. And but if you are in the United States, your state Department of Fish and Wildlife or Fish and Game or Health is also your go-to. And what the herp disease alert system, we have a list of who those people are, and we would help you get that information to them. They are your Ghostbusters for disease, and they can help you get testing. It's important to get the animals sampled and tested pretty quickly, especially if it were to be a B-cell die-off. A rapid response can really help. So there are people out there that can help you with that. And anyone here on this webinar that you see our names there, you could contact us or the disease herp people or your state people. If you're out of the country, then we can also help you there to figure out who the contacts are or provide assistance if needed. And I know there are people that registered for this webinar from 13 different countries. So we're here to help.
Just to follow up too, I think, generally you're all from different agencies and have different rules on what you need to do. So we respect that. And that's part of that plan, I think. But just know that this task force is sort of here to answer questions for specific cases. I think that's mainly the main take home question rather than having a sort of specific answer there. There's another question in the chat though that I'll put forward from Jennifer Buchanan. Can B-shell be shed like snake fungal disease? And the follow-up to that is shedding it, meaning clearing it without being treated.
So I think if I'm understanding the question correctly, sort of asking whether an animal would be able to clear infection without us intervening in some way. Is that correct?
I believe that's so, but Jennifer can clarify. Yeah.
I think the answer is it depends on the species. And so newts, the eastern newts, for example, are one of the species that are most susceptible in the eastern US. And we don't have a lot of evidence that clearance can happen, for example, for that species in the trials that have been done so far. But I think other species do have the ability to become infected and then not show infection later on in our laboratory experiments, whether that's just a really low load that we can't detect or are not infected anymore, is, I guess, an unanswered point or something we can never know. But yeah, it take home, maybe it varies by species, but there are many species that don't seem to be able to clear B-Sal on their own.
Another interpretation of shed might be, the zoospores that are shed from the animal and they get into the environment. B-Sal is unique in that it has a type of zoospore that we call encysted zoospore, and it can survive in soils away from water for a period of time. And that's different than DD, which is really known only to be an aquatic organism. So B-Sal has a capability to be on land for a short period of time, and that shed zoospore could be picked up by something else. So that's another, interpretation of shed.
And then I have another question if we have time in this section for it. So one question from Frederick Huber is, should we be concerned about using pitfall traps that can concentrate animals and possibly facilitate spread?
Yeah, thanks for that question. I think anything that concentrates animals does have the ability to increase transmission. I don't know if others have thoughts here. I don't think it means that you shouldn't use that. I think it is obviously a valuable tool for understanding amphibians and in population dynamics, but I don't know, Evan or somebody else.
I'm just going to say that your hands, nets, anything, buckets that you use to survey amphibians, those also will transmit B-Sal. There's not a concern about B-Sal until we know it's here. Right now, it's not known from North America. So go ahead with your survey methodologies as it is. However, if you're doing intensive research at a site, Gray et al. 2017 has guidelines for research biosecurity, and that includes wearing gloves and changing those gloves between every individual that you handle. And if you are concerned about any of the diseases, that is a biosecurity measure that you could do with monitoring or anything else. In some countries, they require that. In the United States, we haven't gone that far. So
I guess one point to add to that, so Dee was mentioning equipment, and so yeah, B-Sal is not here yet, but we can proactively also be thinking about disinfection if you are going to do pitfall traps in one place and then go and decide to use that equipment somewhere else. Make sure that disinfection is happening between sites, even though we don't know yet that it's here. We can do that as a precautionary measure.
Okay, I think that's it for now. If you want to continue on, we can answer some more questions as we go on.
Okay.
Great. Okay. I think this is me now.
Yep. I will try to anticipate your clicks, but if I don't, just tell me to click.
Okay. So just first, thanks everybody for those questions. We as the North American B-Sal Task Force are here to answer any other questions that you have during this webinar and then afterwards. So there's a bunch of places where our website shows up. You have our contact information, and I think, like Dee Dee said, feel free to contact anybody on that task force, and we'll get you to the right place. So this next section, what we want to do is outline the North American B-Sal Task Force and talk about how we can work together to address this looming threat of B-Sal. Okay, so like Molly introduced, there's a lot at stake here. We have a highly transmissible, lethal pathogen that many scientists expect to arrive kind of at any moment to the US. North America is a salamander hotspot, and so there's concern about amphibian conservation with respect to this B-Sal threat. Salamanders are also the most abundant vertebrate in forest ecosystems, and they're thought to contribute both to terrestrial and aquatic ecosystem functioning. So many of you are here because you care about amphibians, and maybe those are two additional reasons to think about their importance in our systems. So many of you are probably familiar with this white nose syndrome in bats. This emerged in the eastern US. It spread across the United States from where it was first detected in a cave in New York State. It's now made its way all the way to the western US. It's decimated some species of bats. It continues to spread. But what this means is that white nose syndrome managers or folks that are managing bats and their ecosystems, their cave systems, have had to respond in this crisis mode to minimize bat decline since the very beginning. There's a lot of parallels with white nose syndrome, and I work with the Federal White Nose Syndrome Task Force to help them think about disease management and addressing those risks. But they're really in crisis. You're always in crisis mode after a pathogen has emerged. So there's a key difference here with BSAL. BSAL has not been thought to be introduced to the US yet. So we can think, plan, and act proactively and stay ahead of it instead of reacting to die-offs like we've had to do with white nose syndrome and with BD, the amphibian chytrid fungus. Okay, so all of that is really easy to say, right? But disease management problems are complex, they're challenging, and this is certainly the case with BSAL also. They're plagued with unknowns. There's potentially many competing objectives that you and your partners as state managers and wildlife managers have to consider. And thinking about many of these species, like the eastern red-spotted newt, has a very large range. Individuals can roam for kilometers across their life, and so they're the jurisdiction or the agencies that's responsible for habitat or species or population management is fragmented, and so collaboration is really important. All of this together can leave us scratching our head with lots of questions about what can we do, what's possible, what will work. I think, yes, okay. So our hope with BSAL is that we can take all of these complexities and through collaboration, through working together, we can reduce important uncertainties, incorporate irreducible uncertainties or those things that science can't give us answers to into our management planning. We can account for trade-offs with other important resource management objectives that are non-amphibian and non-amphibian habitat related. And we can build and enhance these collaborations to improve proactive and reactive management. And I'm going to talk about the nuance there in a little bit. Next, in fact. Okay, so what can be done? Well, broadly speaking, we tend to think in these steps of working to prevent and prepare introduction and the spread of this pathogen. Then at some point, somebody will notice, we have some surveillance programs, somebody detects a die-off, and we find that the BSAL pathogen is present, at which point we need to have a response. Our expectations that once BSAL is introduced in these populations, that it is likely to spread to other locations and other populations. And so a response is necessary after the detection. Now, one important key point here is that we are in this state where we have an opportunity to prepare populations and ecosystems for BSAL before it is here. So this is what I mean by proactive management. I'm going to take a hot minute here to make the distinction that proactive management does not mean simply planning for a response. It's not planning for the eventual detection. What are we going to do once we have a detection? What we're talking about here is we have an ability to implement management actions, making a decision to irrevocably allocate resources, to change populations or the ecosystem in which our amphibian populations occur, to reduce the susceptibility to the negative effects of BSAL. So this is optimal in disease management. Folks talk about this in human pathogens, wildlife pathogens, and captive animal populations. But being proactive is really hard to do because oftentimes we can't anticipate the threat before it arrives. But here we have that opportunity. Okay, now there are different dimensions of what we're talking about here. To be proactive, we can work to prevent or minimize entry. So this is represented by the salamander rule that Molly talked about. Also, folks are working on developing these healthy trade programs to try to reduce or minimize the probability that BSAL is going to be introduced into populations in the US. Next, we can work with resource managers to frame their management decisions identify what actions could be implemented before B cell is even detected. And through this process of collaboration between science and management, we can identify optimal courses of action to minimize B cell declines and work to develop and test implementable management strategies. So figure out what will work and implement that. On the other side, right, after we have a detection, if we haven't implemented proactive management, or even if to enhance the capability of proactive management to reduce the susceptibility of populations, we can plan for improved reactive management after B cells detected. Okay, in this case, we can work to facilitate rapid response. And so there's working groups within the B cell task force that are working on categorical exclusions for low risk management actions. We can improve governance structures that will improve collaboration. So in the middle of a crisis is not the time to start thinking about who you need to call and who you need to work with in order to respond to a threat. We can do that ahead of time and proactively plan. We can implement improved biosurveillance networks that give us the best chance of early detection of an outbreak of B cell. Okay, so all of these things, this proactive mindset, thinking about developing actions, working with managers, working with scientists to try and figure out what to do ahead of an introduction, while also keeping an eye on planning for an eventual detection. This mindset is the heart and soul of the B cell task force. And we'll get into the nuts and bolts of this shortly. But first, to make sure that everybody is still awake out there, we want to ask you this question. And Molly will have to explain how you respond to this. But what we want to hear from you, and this will help the B cell task force in the work that we do going forward, is to understand what you think the major challenges are to your ability to respond to a B cell detection. And I'll hand it over to Molly right here.
Yeah, thanks, Evan. You should be able to see, I think Carrie will post in the chat, this poll.ev.com link. But if you go there, you will be directed to a place where you can input either a word or a phrase as something that you think about when you're thinking about major challenges, and that will appear here as a word cloud. You can enter things multiple times or multiple ideas. You don't have to type it all at once. It'll continue to auto-populate as folks do that, so we can stay here for a little bit and let folks respond.
Molly, I'm not sure if this is working because I've just typed in a response to test it and I don't see it popping up. You're also muted.
Are folks not seeing what's popping up? Oh, I can see it on my end. Interesting.
It says it's maxed out of allowed answers. Poll is full, is what the chat's saying.
All right, well, I'll go ahead and stop sharing and I'll reshare and see. I can also, I don't know what's best for moving forward. I can read some of the responses, which maybe is better than resharing.
I think that our intent here was to use these responses to bring these back to the B cell task force and to think about how the B cell task force can come up with a response to these and which working groups are responsible. So if we don't have time or we can't figure out how to share that, I think we can summarize the response back out to this group.
Yeah, I will attempt here once to do the reshare and see. How about now?
Nope.
Nothing.
No. What can I ask what you are seeing?
Just the blank. What do you think? Just the blank question slide.
the major challenge is
Okay.
technology, turns out.
Yeah.
All right.
All right. Well, some of the words that are displaying are communication, information, cooperation, agencies, symptoms, training, availability of funds, resources, diagnostics, response, limitations of funding, organizational structures, interagency collaboration, I presume, politics, clarity, tools. So that gives you an idea of what words were submitted. Apologies for the technology not working.
Excellent. Thanks, Molly. Okay, so this is as we expected, a great lead in to our next slide, which is the introduction or the introduction to the BSAL, North American BSAL Task Force. So this technical advisory committee is an interdisciplinary group working from multiple different angles to help prepare and plan for BSAL emergence in North America. This group was initiated at an international workshop that we organized in 2015. The task force is a collaborative network that has members from federal and state agencies, academia, and non-governmental organizations across North America. There are currently 8 working groups, which are here at the bottom. And the working group leads, in addition to a few others, form this technical advisory committee, which meets monthly and shares updates from the work of the working groups and any new news on BSAL. There's this executive oversight committee here above, which provides opportunity for receiving outside direction. Some of those members sit on this technical advisory committee. So we have communication both from the individual working groups that are working intensively on these different parts of this BSAL kind of problem, and the oversight committee that's giving us some context on the world out there. Next slide. Okay, so like I said, the BSAL working groups, this is the real heart of the North American Task Force. Each group is working with their own goals and objectives, and these are derived from our interactions with the amphibian conservation community, resource managers, and other kinds of decision makers. So we really, the decision science here is one of the working groups that we'll hear from today. Really, the North American BSAL Task Force, we really want to be focused on conducting research, conducting surveillance, thinking about communications and outreach, that all help to inform management and amphibian conservation decisions, hopefully that are proactive, but also are prepared in case BSAL arrives. So as you can see from the diagram, all of the working groups are interconnected and are designed to interact and feed off each other to effectively address this BSAL threat. So we're not going to walk through all of these working groups today. If you're interested in the work of any one of these in particular, again, let us know at Salamander. Look for our contact info, and we'll share that we'll share contact info for individuals at the end. We will outline the work of these 3 working groups: decision science, response and control, and surveillance and monitoring, that directly intersect with on-the-ground management. Okay, so one of the major collective efforts across these working groups has been development of these products that we want to highlight and share. So first, here on the left, this is our strategic plan. This outlines the goals of each working group individually and identifies connections among groups that form the BSAL Technical Advisory Committee and the task force. The second in the middle here is this implementation plan. So this steps down the strategic plan to provide more specific work plans for each of the working groups, again, alone and together. And then the third, Molly and Dee Dee talked a little bit about this in the first part of our presentation here. This is our response plan. This helps navigate how to detect, how to confirm a BSAL detection, and offers initial guidance for how to develop a response to a BSAL detection. And importantly, these products can all be found again at this website, salamanderfungus.org. And if you're interested in anything you find there, there's contact information for lots of people there you can follow up with. We also produce annual reports for each year, and we're assembling the annual report for 2022 right now. So that'll be up on the website soon. Okay, so the first working group that I want to outline is the Decision Science Working Group. The current lead is Molly Bletz. So as you might have realized at this point, one of the main goals of the BSAL Technical Advisory Committee and the task force is to assist with development and implementation of proactive management. We don't want to wait for things to die to start identifying and implementing management actions. And in fact, there's lots of research that's going on that provides some clues into what might be reasonable management responses. So this group has worked to forecast the risk of introduction in collaboration with other working groups and other folks outside of the Technical Advisory Committee. We find that there's regions in the US where we expect eventual introduction of BSAL in North America. And Molly showed that map that Dan Greer, the publication that Dan Greer led. These places are especially the Mid-Atlantic, the Southeastern and Northeastern United States, and most of the West Coast. So lots of areas of the US are at high risk for introduction and effects of BSAL. So taking this information about the threat, trying to think about the research that's out there, and thinking about the particular management problems and context, the Decision Science Working Group works to frame and solve management problems. So if you as a resource manager are unsure what action you should take to reduce BSAL risk, or what action might be useful in response to a detection, you can partner with folks in this working group, and they'll help with that. Additionally, this working group can also help identify research needed to improve management and guide data collection, and use a whole suite of tools for solving complex decisions that come from the world of decision science. I just want to show some examples of current decision support. We don't have time to get into these, but these are active. We're working on these actively. We're working to support preparations in response to BSAL multiple ways. So there's two case studies here that we're working with now. One is a high elevation New Mexico endemic salamander in Eudis hardii, which is highly susceptible to BSAL. BSAL hasn't been detected there, but we're working with the US Forest Service, the Mescalero tribe, the state of New Mexico, and Fish and Wildlife Service to think about how to implement proactive management in this high elevation system where there's lots of uncertainty about the species, ecology, and there's lots of competing objectives. So the Forest Service manages for multiple use. Salamanders conservation is just one of the considerations there. So it's a complex and challenging decision with lots of uncertainty. We're working with Fish and Wildlife Service refuges in the northeastern US to identify the optimal proactive and reactive management strategies that they might take to reduce the risk of BSAL to the eastern red-spotted newts that are present across the northeastern US. Along the way, we're developing models for disease management, and those models that we are developing are particularly relevant to these two contexts, the New Mexico salamander and the northeastern refuge salamanders. And through this process, we can identify those most important areas of research, those research questions that help to better inform the decision. Molly, do you have anything to add?
Nope, I just unmuted to talk through the polls here if you're ready for that.
Okay, yeah.
Okay, yeah. So before we talk about Evan, I'll talk about the next response and control group. We've shifted the polls to a Zoom poll platform. I believe maybe Carrie can unmute. But so the next question we have just is related to the response planning that folks have, and whether you have, Carrie, do you know if, before I say this question, make sure that we actually have the poll here.
I have the poll ready. You can transition to the next slide.
Oh, okay. I should go to the next. So here we're interested in basically whether you have a contact list of if you did have a BSAL event, a BSAL detection.
have just about half the participants responding so far, so we'll give it a little bit more time.
All right. Does everyone see these polls as they go? Yeah. Okay.
For some of you who might be on the web browser, you probably won't see the link for the Zoom poll. Okay, well, it looks like responses have slowed down a little bit. We've got a lot of folks who have answered, and I'm just going to share the results.
All right, so yeah, we have, let's see, 26 people who have said that they have a contact list. That's really great to hear. There's a subset of folks who answered no, that don't or are unsure. And so that perhaps is an opportunity to get in touch with the response and control group about ways to think about communication planning and sort of how you can prepare that contact list or work within your agency to develop that. And we are, of course, here to help in that process. All right. So we had a second question. In theory, we can't advance this. There we go. Poll question here about if a B-cell disease outbreak is suspected to have caused mortality in a wildlife management area, sort of select all that would apply in sort of how you would respond to that event.
Seems like we have a really awake group today. Lots of responses are rolling in. So thank you for participating. This is really important information for us as we're looking forward and trying to plan for additional projects and resources to help support you all.
I will say one thing about the two middle choices here: implementing responsive management such as quarantine of the site, and implement responsive management such as treatment of surrounding sites. So there, the quarantine or treatment, those are just examples of single site actions, or if you have a concept of how you might respond at the larger kind of landscape scale. Do you have those collaborations in place? Are you responsible for multiple populations? Those kinds of examples.
Is everyone able to see these responses now? Okay. Do you want to talk about the responses, Evan?
Go ahead, Molly.
All right. So we have about 50% saying conduct additional surveillance, 45% that have said implementing responsive management, 20 such as quarantining, 24 with treatments of the surrounding sites, others saying that they don't have a plan. And so I think this is a place where a lot of working groups can also sort of step in and help as needed. If you're interested in response planning or planning and working out those decisions, the decision science group that Evan and I lead can help frame those decisions and think about what you can do and when you can do it, not only reactively, but proactively. And also the response and control group and thinking about sort of using that rapid response template document that we have for how you proceed through a detection event as well. So I will.
Yeah, I'll just pause for a second and acknowledge one thing, that the wildlife diseases, B-cell maybe in particular, this is a complex decision, right? And there's lots of things that make that complex. And just to echo what Molly was saying, the BSAL Technical Advisory Committee is here to help. There's lots of expertise there. And so definitely reach out to us, particularly if you're in that do not have a plan, but desire a plan kind of category. Okay, the next working group that we want to highlight is this response and control working group. They're working to facilitate and streamline our ability to respond to B-cell emergence. This includes that rapid response template that we showed that has guidance on how to respond to die-offs, uncertain causes. So if you find a dead amphibian or a bunch of dead amphibians, and you need some confirmation on whether or not that is B-cell. Again, this is found on the salamanderfungus.org website. This working group is also working on efforts to identify and streamline administrative approvals in order to take action. So this is something that probably is jurisdiction specific and can vary greatly among states. And it would be really wonderful if we could work with more state agency folks, could get them involved in this group to help make sure that the work from this response and control working group is actually useful to individuals that are making those kinds of decisions. There's lots of parallels with invasive species and invasive species management, and we have some experts in these response and control working groups that works in these other systems that are bringing those expertise to developing this for B-cell. So examples of this are categorical exclusions, the NEPA EAs and EISs, EPA Section 12, but this is a lot of administrative policies, and this working group needs help navigating these rules. So if you have expertise, we ask you to contact your Laura Sprague to join that working group and move forward. Okay, the next working group that we want to talk about is the surveillance and monitoring group. This initiated and coordinates a wide-reaching, ongoing, and coordinated B-cell surveillance program. Currently, this has 29 participating locations in surveillance through this Student Network for Amphibian Pathogen Surveillance, or the SNAPS program. One benefit is that students benefit from real-world research, and it additionally contributes to our ability to detect B-cell quickly. So really working on the collaborative effort of universities to get information that's useful for detecting and responding to B-cell. The current group lead is Ali, and you can contact him at his the email address here. This program is currently brainstorming strategies for funding to make it more sustainable and to support new partners. So there's other groups that are in other institutions that are interested in participating, and drumming up more funding support, I think, is key to expanding and continuing the SNAPS network. One model being discussed within and outside this group is whether partnerships with state agencies where institutions are located could help provide some supporting funding for this. And so contact Ali or again, anyone in the task force if you have ideas for or interest in this surveillance and monitoring group. Okay, data generated during SNAPS is deposited in this amphibian disease portal. The website is here, amphibiandisease.org. It's a public repository that everyone can use and access. This is developed and curated by the data management group. And here we show just an example. This is a map of the SNAPS partners. I was looking at it yesterday, and there's a lot of good detail on sampling that's occurred from the SNAPS program, but also other surveillance data. So it's a good resource to see what the current level of information is for your particular location. And also if there are gaps in sampling or spatial sampling that you or partners might be able to contribute to. So again, look at this amphibiandisease.org website and contact Ali or anyone on the task force to connect you with this resource. Okay, so we didn't have a lot of time today. Our two goals were to give you some background on B-cell and an introduction to the B-cell task force. We didn't have a chance to highlight the work of all the working groups today. And like I mentioned, there's lots of stuff going on behind the scenes, and we want to make sure that we are relevant and responsive to this B-cell conservation problem. But we do know that working together can better allow us to address and proactively manage for B-cell and there are a bunch of ways that, that we can take action. So first, as we discussed, B-cell is a different beast than BT, but the symptoms can be similar. So if you see something, if you see sick or dead amphibians, say something, reach out and let folks know that you have observed this. That kicks into action a whole bunch of processes that help alert the right people, that help to bring in this B-cell task force and to be able to respond very quickly to a detection. There are multiple opportunities for proactive planning and for proactive management that can really shift the narrative of conservation from not only reacting to die-offs and detections, but being proactive and reducing the risk to our native amphibians. So let's capitalize on that together. Again, reach out. There's someone who can help. This task force is also strategically planning and developing tools to address B-cell. So please, at the end of this, we'll have an opportunity to ask more questions, but as you think of them later, reach out to this group with questions and recommendations for how we could help. We're eager to know how we can better meet your needs and we're interested in expanding our team if you're interested in contributing. There's always a place for folks that want to help respond to this threat and address amphibian conservation. Okay, so I've said this probably 8 or 10 times, and I, working with this task force, they've heard me say many, many times, but one thing we want to consistently emphasize is that we are in an excellent position right now, in an enviable position in terms of wildlife disease management, to be able to enact proactive management instead of reacting. We have a lot of tools and guidelines that we've developed and we've used science for so far, but we need folks like you to know that they exist and how to use them. We also need your input on what could help you better prepare and manage for B-cell. So right now, this opportune time to strike, as a focus on One Health at the national level means there are potential funding avenues for disease response planning, disease active proactive management. And in addition, thanks to the American Rescue Plan Act funds, the regional Fish and Wildlife Associations are in the process of hiring regional wildlife disease coordinators, further providing opportunities to proactively address this disease threat. This Recovering America's Wildlife Act also has passed the House and may go to the Senate soon. And if passed, it would provide $1.4 billion each year for state wildlife agencies to manage species of greatest conservation need. And this is a status that many salamander amphibian species fall under. So this funding may be able to be used for disease management actions, among other conservation actions for amphibians and other wildlife. These state wildlife action plans, or the SWAPs, which are in revision that are due in 2025, are useful roadmaps for that funding. And currently state agencies in the process are beginning revisions of these SWAPs. Okay, so just to sum up, as we mentioned throughout this presentation, we want to know and address your needs. The B-cell task force is here to help respond to this threat. So perhaps you're a manager looking to prepare and make informed management decisions for B-cell. We encourage you to contact the Decision Science Working Group. They have expertise to help frame and solve these complex management problems, and they'll be happy to help. We also want you to feel welcome to join the task force. You have expertise, you have experience that could be useful for our group. If you're interested in developing guidance and policy to facilitate rapid response to B-cell, contact the response and control group. If you're interested in contributing to surveillance and monitoring, there's a working group for that. And in general, if there's anything that you see, any of your expertise or experience that you think you could lend to this effort, please contact us. And I believe that this Scan Me thing will work if you, Molly.
Yep, so that QR code will link you to, I think, what Carrie has also shared in the chat, which is a Google form for the follow-up to this that has an opportunity to answer a question of, we would like the task force to contact me, or you'd like the task force to contact you, or you'd like to get involved in a specific group, and then we can coordinate and get you in touch with the right folks.
Thanks, Molly. As part of registration, we also ask folks to write in questions that they hope are addressed, and we use those to help guide this presentation. We know that all your questions haven't been asked, so this is the opportunity now. I think raise your hand or send questions in the chat, and we'll do our best to respond to them here.
So I'll jump in with some questions that have been asked just as people are thinking of other questions. But one of them is tying to the surveillance slide that you had. It's from Jamie Utes, maybe is how you say your last name. Are there organizations that are taking in opportunistic samples from the field from folks across the country, or that offer funding for equipment and costs around testing? And Aubrey Hill mentioned, of course, SNAPS, but that probably is more focused on university and student sampling, if that's right. And so there are questions about, I think probably for many people here interested in surveilling, and how do they pay for that?
So oh, go ahead, Evan.
I was just going to say there are regional kind of disease collaboratives where you can work through your state herpetologists or state wildlife agencies to submit detections. There's opportunity to do analysis there. Didi, maybe you can add to other.
I was a little bit distracted, Evan, when you were talking right before the SNAPS slide came up, and I wasn't sure that you echoed that USGS had done over 10,000 samples across the United States before SNAPS got going. So there has USGS in the Department of Interior had done pretty comprehensive sampling to see if BSAL is already here. And so there are organizations that have done some, but at this point, the universities are getting involved. And then there's private individuals that are doing sampling as well, because you can publish your results of no detections, because we want to get those in the database. There is a database of all the projects ongoing for BSAL at the data management working group has a Kitrid database online of all the data for BD and for BSAL. So you can go there to see if there are ongoing projects that are relevant. So anyway, I just wanted to put that out there. The only other thing for BSAL is if there is a die-off and animals are submitted to a laboratory for diagnostic testing, then they're looking to see what suite of pathogens they have. And so, and the most recent ones I know from the Herp Disease Alert System were Ambystoma salamanders, tiger salamanders in particular, that were submitted for testing. And it was ranavirus, it wasn't BSAL leading to those die-offs. But if they would also look for BSAL. So that's a reactive, not a proactive surveillance.
And I would say too, I mean, they mentioned the diagnostic working group. So I'm the chair of that group. On our website, there are a list of diagnostic labs that can perform those. But even more importantly, again, because many of you are tied to agencies, you may have sort of internal or different ways you can send sites or samples to. And we're more than happy, that group to help work with those folks. So, I mean, we can run samples, but also we can let people know how to run the samples. They're pretty standard PCR type tests that if people are already doing a bunch of different sampling for other pathogens, we can sort of help guide them on the, sort of best practices there as well.
In monitoring the literature, there was a recent paper about surveillance in Mexico, and they did not detect BSAL in that paper. So yeah, and those sorts of information, if once things are published, we can get them uploaded into the data management system so everyone can see it.
The other questions, there's two that are sort of linked, and this is about BSAL in Europe. So one was asked earlier about just dynamics of BSAL. So in Europe, where BSAL has been found, has it reduced the populations to zero, basically extirpated them, or has there been a plateau where there are resistant individuals? And And another sort of follow-up just from from Nate is sort of how rapidly we think B-cell is spreading once it shows up at these new European sites, how quickly it colonizes other nearby sites on the landscape. So I think trying to gather some information from folks in Europe about how it's spreading there and what the dynamics are, and we can sort of use that information here. Thoughts on that?
So I can comment on some of it from the well-studied and well-followed up populations. I would in Belgium and Germany, I know there's repeated that it's pretty big crashes of the populations, but I do believe they still find a few individuals. It's not always known if that is a individual who has persisted versus a new colonization, sort of a recolonization of a given site. I think European landscapes in a lot of places where B-cell has occurred are pretty fragmented. And so they have an outbreak site that can be very close to a site where there's salamanders that are completely fine. And it's not jumping those short distances because perhaps there's a barrier like a big road or something that just is minimizing animal movements. There have been, I would say, big jumps, if you will, of sort of outbreaks and die-offs in Germany. Suggesting that that's probably not salamanders that are moving it, but other things. If your natural habitat, I'm sure that there might be folks in from Europe, so please feel free to raise your hand if you have other information you want to share here. But I think the transmission dynamics across the landscape are very complex, and sort of understanding how it's gotten where it's gotten is not fully fleshed out. But it can seem to move quickly and in not a specific pattern, if you will, and jump around. Evan.
Yeah, maybe one of the nuances of the question was like learning from the European system and how we translate that to the US. And so, we've doing the surveillance and research on European amphibians and the response to B-cell, it gives us some kind of expectations of what may occur in the US. But like Molly is saying, it depends on a bunch of stuff. And so, using that information to think about the management decisions that you might implement proactively or how you might plan a surveillance design or things like that. These are all very complex questions that I think there's not a general answer to. But reach out to the TAC and somebody can help you think through those.
And there's an interesting life cycle. I mean, these amphibians have complex life cycles sometimes, and there is a terrestrial component of the population not at the pond at any one time. And so you may have some animals that aren't being infected that are out there, or that be, the next year when breeding occurs, they emerge from their terrestrial haunt and come to the site. And if B-cell is still there after a die-off, they may get reinfected. So these are complex dynamics that we need to bring into play, and only just a handful of examples of heavy-handed treatments are known, I think, at this point. But there is one example of hope for eradication of B-cell at an isolated site.
The last comment I'll make about Europe, there is a website called bcelleurope.com. We have a link on our site as well for that. And we, of course, are in communications with researchers behind that initiative as well. But if you're interested in details about what's happening more real time in Europe, that is a great resource that is kept pretty up to date.
So Jake, can you answer the question about eDNA sampling for B-cell?
Yeah, it looked like there were two samples, one or two questions. One was about cost. And again, that varies. How much does it cost to run a sample? That really varies, of course, within the different agencies, of course, structures that you have. I mean, it could be free, if you will, I suppose. But generally the actual cost, a lot of that is overhead and knowledge and equipment to run those things. The actual supplies for it, I think you could probably get it to be as cheap as $10 a sample, but more typically it's probably $20 to $30 when you sort of incorporate all those other costs. Again, there's different methods. I run a lot of samples for graduate students and find lots of cheap solutions, frankly. So things like pooling samples, if you want to sample large numbers of animals in one sample, you can have sort of lots of representatives in one sample that you're, paying that much money for. So lots of solutions again. And if there's specific questions from folks, we can talk about those and field those as well. In terms of eDNA, again, yeah. So with that as well. So oftentimes the sampling is done for B-cell via swabs, since it's a skin infection. That seems to be a nice non-invasive way of getting information. But of course, another one is just sort of using water or other things, soil and whatnot. And again, it sort of has all the problems of eDNA. So contamination is a big concern there, and detectability. So when you're swabbing, a zoospore off of a salamander, that is a pretty good sample versus, filtering so many gallons of water to hope you get a zoospore can be more difficult. So I think it's an effective approach if you have trouble getting your hands on the animals, depending on where you're at and what the environment is. And allows you to sample for lots of other things simultaneously. So that's a approach a lot of people use. But generally, I think, again, I would stress, as been said before, if you have a sense that there's a die-off occurring or animals are sick, the best thing you can do is to take that animal and send it to someone. So as we highlight in the document, it hasn't really been mentioned here, but just because we get a positive PCR result doesn't really mean it has B-cell, because there's lots of false positives and problems that we have with that. So really, I think I'd emphasize from a diagnostics perspective is try to grab the animal so we can send it to a vet to do pathology and things like that. But again, these are all important things in terms of surveillance. So testing for the DNA of those is great first indicators.
So there was a comment, not really a question, from Antonio Salas in Peru, and he commented that it's hard to get attention for amphibian disease issues there, and this is a common denominator worldwide, that amphibians don't get as much attention as other wildlife. And I would like to think that maybe the North American B-cell Task Force could raise this in our next meeting to see if there's an opportunity for an international working group and provide some support for other places to raise the issue. And it may be beyond B-cell, but B-cell, of course, is the central issue here.
That raises a good point, Dee Dee, and that's actually come up in some of our Amphibian Survival Alliance meetings of how do we coordinate, across these different regions for amphibian disease management or amphibian disease mitigation planning. And so I bet that network also would be very interested to engage with such a effort.
We do have international members of the research working group, for example, and maybe some other groups that I'm not quite sure of, but we might be able to provide some resources and some assistance. Not what we've been doing would work everywhere, but there might be something that we can help with.
Yeah, the Decision Science Working Group also had some international membership for a number of years. And all this is really useful, right? Because we can learn about research, we can learn about management decisions and context, we can learn about ways other folks are conducting surveillance in different parts of the world, and that can improve everybody's kind of planning and response. So yeah, we'll bring that back to the TAC in the next working group call.
And the other question or sort of comment is actually almost contrary to that. So some folks aren't interested in amphibians, others are maybe interested too much. So they have a lot of conservation work focusing on public lands that has an urban and rural interface, which presents challenges but opportunities. Very well-meaning members of the public who care, but almost care too much. They want to be involved, but can contribute to disease spread. And ideas for developing a plan for public engagement that works with their good energy while also providing protections for the resource.
Yeah, I think this is really, this question or comment forms kind of the fundamental structure of the Technical Advisory Committee, and that we have all these working groups, right? they're all centered around thinking about management and those management decisions. And so how do you balance when there's an ecosystem that has amphibians is one component of the ecosystem, but there's some other resource that's more highly valued. Or when you have folks from the public that are interested in contributing, those are really involved and interested stakeholders. How do you have them contribute to this decision-making process for amphibian conservation, of which BSAL is a motivating aspect right now? These are the kinds of questions that the BSAL task force is working on. We think that it starts with, because this is all conservation management focused, we think that this starts with the folks that are making those conservation management decisions, helping them to think about how to incorporate those stakeholders, having those stakeholders identify those managers or decision makers that need to be involved, thinking about how to incorporate amphibian objectives into other kind of resource management decisions. This is all kind of the complexity of the decision that this task force is working towards.
I think the communication, in terms of even just communicating with the public, the communications group, is looking for opportunities to develop materials that might help with communicating about BSAL. So if there are community engagement projects or community science projects where you want this to be part of the conversation, perhaps working with that communications and outreach group would be a good place to sit or to connect with as well.
And if I could jump in really quick, I was just going to mention too, some of those disease resources by the Northeast Park that is on our park website, like the disinfection protocols and all of that, that's really helpful. So working with master naturalists and volunteers out in the field, one of the things we always try to hit home is the importance of biosecurity, cleaning your boots and your equipment before and after going to every different site. And so it's not specific to BSAL, but it also helps with all sorts of other amphibian and other types of diseases that might be carried on equipment. And it just gets them in that mindset. So I think kind of integrating that into your programs and making sure you have some of that there will get them thinking about the biosecurity protocol, no matter what disease you're dealing with.
So I'm looking at the question from Rich Staffen. Do you see a role for salamander or vernal pool monitoring to help monitor for BSAL? And I think linking disease surveillance to amphibian surveillance is a really good idea. And it's not specific to BSAL. It would be for BD, but not for BD generally. Now we're interested in what strain of BD is out there, because the different strains of BD have different virulence. Some lead to disease and some do not lead to disease in the various species. So I think it's a really good idea to link an uptick in amphibian surveillance and monitoring with disease surveillance and monitoring, or in this case, pathogen surveillance and monitoring. And I would do the suite of BD strains, BSAL, and ranavirus in particular.
I mean, that's a golden opportunity. Anytime there's somebody out there looking around and they're not seeing something is really key, because right now we have lots of information on detections. It's relatively less clear where people are not seeing amphibian pathogens. And so, yeah, definitely.
Okay, that's all the questions I see. If folks have other questions, feel free. I think at this point, if you want to pipe in and ask too, that's fine.
Yeah, Carrie, do you want to open up the muting so people can talk if they want to?
Everybody is now able to unmute themselves. So we've got about 5 minutes left of our allotted time. And we will be collecting all of these questions and trying to summarize them as well.
I do see a hand up. Yeah.
Real quick,
I have too many screens, but please feel free to unmute and talk. Yeah.
Katie Weil from Oregon. When we do monitoring and surveillance, there are many agencies that are on the smaller side that don't necessarily have the resources to pay for that testing. We are out to be eyes and ears, and we can collect information, but I think it would be interesting to find alternative funding resources to support that testing, whether it's somebody can pay for shipping, somebody else can do the lab. We have to go to 2 labs. So I'm just putting that out there. Thanks.
Maybe with this new legislation that's coming down the pipe, if it
Right.
gets signed.
Yes. Thank you.
Yeah, this is where we can think proactively too about resources like Recovering America's Wildlife Act. It will include disease resources, but it has to be, you know, a priority for the state agency. And having that type of information put into the state wildlife action plans will make it easier to justify using that funding for those types of resources and actions.
Also thinking about other kinds of information that can be collected. So some folks that I talked to, you know, they ask about, like, and I think Molly, you alluded to this in the beginning of the presentation, is like, well, amphibians, right? They have BSAL, or they have BD, they have rhinovirus. Like, we expect to come up to a pond and find a bunch of dead amphibians, right? So, you know, changing that, thinking about, well, the every dead amphibian every set of group of dead amphibians that you see is potentially interesting, and so maybe some way of having better reporting of just the observation of sick or dead amphibians, and then this extra step of, well, what is causing that, right? Is it BSAL? Is it BD? Is it rhinovirus? And really, if the ultimate goal is amphibian conservation, sometimes the management actions don't depend on knowing what the pathogen is, right? The management might be similar. If it's one pathogen or other, you just see it die off. Certainly knowing about BSAL being present in the US, that's a new threat. We expect kind of dynamics of BSAL to be different than BD and rhinovirus in terms of how it might spread and cause declines in populations that are different and distinct from rhinovirus and BD. But even knowing about these die-offs is useful.
And I would just add in, I'd encourage you all to talk to people in your different groups and agencies. I mean, I think obviously we're working hard to talk to folks on our end to reach out and get funding, but probably the more voices that are heard from lots of parts of the country would create more of a, you know, prioritization. There's limited funds, of course, and there's lots of stuff to think about. But if you feel like this is important in your particular region, then I think using your voice there is only going to help. So I encourage everyone to do that.
All right, well, I want to thank everyone, particularly our 2 speakers, Molly and Evan, and then certainly everyone on the BSAL task force who helped with this presentation, who's been helping behind the scenes and running things, and also for all of you for staying with us and staying engaged and helping with this information. This, all of what you've submitted with the polls and with the Google form and all of that, we're going to be looking at that and helping to figure out essentially how we might be able to work in new directions or new products and things like that with the North American BSAL Task Force. So I'm going to put a link in the chat really quick. This is to the form if you want follow-up information. That includes the monthly, signing up for the monthly park newsletter. We'll have one going out next week. We also are going to start putting together a list for folks who just want information on herp diseases and with the BSAL task force. Also, if you're interested in more information from certain task force working groups, or if you're interested in joining, if you can fill out that survey, that will be really helpful. So this webinar has been recorded, and once captions have been edited, it will be up on the Park Disease Task Team website and also on the Park YouTube channel, and I will email it out to everyone who registered. So thank you again, and I hope you all have a wonderful weekend, and please keep in touch.